Hello everyone, I hope all of you are doing great. So today let us start with a very small topic which is from the chapter Cell, the Unit of Life, and the topic that we are going to start is the nucleus. The topic that we are going to start is nucleus. Okay, so if you look at cell, nucleus is actually present in which kind of organisms nucleus is present? Nucleus is actually present in eukaryotic organisms. Okay, nucleus is actually present in eukaryotic organisms. So nucleus is absent in prokaryotic organisms. Okay, so what is nucleus basically? Nucleus is a double membrane cell organelle. It is a double membrane cell organelle. So we have, in our entire cell biology topic, we have three cell organelles which are having double membrane around them. The first cell organelle is nucleus, which is double membrane cell organelle. The second cell organelle which is double membrane is the chloroplast, and the third cell organelle which is also having two membranes around it is the mitochondria. So we are going to discuss about one cell organelle which is the nucleus, which is double membrane. Now this nucleus was first described by a scientist called as Robert Brown. So Robert Brown was actually a Scottish botanist. So he was the first scientist who actually described the nucleus in the year 1831. In the year 1831, Robert Brown was the scientist who actually described the structure of the nucleus. Now, along with this, we have one more scientist which NCERT mentions about, the material that is present inside the nucleus. The material that is present inside the nucleus was stained by using basic dyes. The material that is present inside the nucleus is stained by using basic dyes. The material inside the nucleus was stained using what dyes? Basic dyes. And that stained material is named as chromatin. The stained material is named as chromatin by a scientist called as Fleming. So two scientists, we'll be coming across two scientists in the entire paragraph of the entire content of the nucleus. We'll be coming across two scientists in the NCERT. First scientist is Robert Brown, who actually described the nucleus in 1831, and the material that is present in the nucleus was stained using basic dyes, very important, using basic dyes, and it was named as chromatin by a scientist called as Fleming. Now, what is the meaning of this word chromatin? If you look at chromatin, chroma is actually color. Chroma is actually what? Color. Tin is actually thread. Tin is actually thread. So chromatin, it is talking about a colored thread. Colored thread, which is actually DNA, which is actually the genetic material. We'll be talking about it in a little while from now. So these are the first few sentences of the NCERT. The first thing is, we are talking about a double membrane cell organelle which is nucleus. Nucleus is found in eukaryotic organisms. Okay, little biological classification we'll bring in here. What are the four kingdoms which are having nucleus? The first kingdom in which nucleus is present is the Protista, kingdom Protista. Then you have kingdom Fungi, in which true nucleus is present. Then you have kingdom Plantae, in which true nucleus is present. And then you have kingdom Animalia, in which true nucleus is present. So these are the four kingdoms in which you have true nucleus. And the only kingdom in which true nucleus is not present is the kingdom Monera, where true nucleus is not present. Means you don't have genetic material, is not surrounded by anything like nuclear envelope in the case of members of whom? Monera. But we have 4 kingdoms in which 2 nucleus is present, that is Protista, Fungi, Plantae and Animalia. Now, based on the number of nuclei, how many nuclei are present in the cell? So when I am using the word nuclei, I mean I am talking about more than one nucleus. When I am using the word nucleus, it means it is single, one nucleus, single nucleus, it is nucleus. When I am talking about more than one, it is nuclei, plural form is nuclei. So generally most of the cells have only one nucleus, single nucleus is present in most of the cells. So if single nucleus is present, one nucleus is present, single nucleus is present in the cell, this is called as uninucleate condition. Uninucleate condition or mononucleate, uninucleate, single nucleus is present. Sometimes you have more than one nucleus. Now let us take for example, there are 2 nucleus. If there are 2 nucleus, this condition is called as binucleate condition. 2 nuclei are present, what is the condition called as? Binucleate condition. Now binucleate condition, where do you find binucleate condition? But let us take for example paramecium. Let us take for example paramecium. Binucleate condition is present in paramecium. Next, if you move forward and if you are looking at the sexual reproduction in fungi, if you look at the sexual reproduction from fungi, in the members of Ascomycetes and in the members of Basidiomycetes, after plasmogamy, plasmogamy is the fusion of the protoplasm. After plasmogamy, there is a stage which is called as the dikaryon stage. So during this dikaryon stage, the fungal hyphae is going to have 2 nuclei per cell. So dikaryon stage of fungi, what karyon stage? Dikaryon stage of fungi is also binucleate. So wherever you are having more than one nuclei, if 2 nuclei are present, it is called as binucleate condition. For example, paramecium and dikaryon stage in the case of fungi. The next one is the multinucleate condition. The next one is the multinucleate condition. Presence of more than 2 nuclei, or even more, you call it as multinucleate condition. But a multinucleate condition, where do you find this multinucleate condition? Multinucleate condition can be found in tapetum. What is tapetum? Tapetum is the innermost layer of the anther wall. Anther wall, innermost layer is the tapetum. So anther wall, outermost layer is the epidermis, followed by the epidermis you have endothecium, followed by the endothecium you will be having middle layers, and the innermost layer is the tapetum. So tapetum is going to show more than 2 nuclei. I mean, not every cell of tapetum has to show more than 2 nuclei, but generally a polyploidy condition is present in tapetum, multinucleate condition is present in tapetum. So one example of multinucleate condition in the case of plants is tapetum. Okay, so these are some conditions where nucleus is present, 2 nuclei are present and more than 2 nuclei are also present. Now, there are some cells which are living but do not have any nucleus. There are some cells which are living but do not have any nucleus. Nucleus is not present in them, but they are still living. So those cells which are living but do not have any nucleus, examples of those cells we will be seeing. The first one is RBC in the case of mammals. RBC in the case of mammals, but it is enucleated. Enucleated means does not have nucleus. Similarly, sieve tube cells in the case of phloem. Sieve tube cells in the case of phloem. So here this is also enucleated. It is living but it does not have nucleus. So sieve tube cells in the case of phloem and RBC are one of the few examples of cells which do not have nuclei or cells which lack nuclei. Okay, so this is about the nuclei based on the number of nucleus or presence or absence of nucleus. So next we will see the types of nucleus based on the types of cells based on the position of the nucleus, where exactly nucleus is positioned. Based on that, we'll be seeing different kinds of cells. Now, just to begin with, if we take an animal cell, we know animal cells are devoid of vacuoles. Okay, animal cells are devoid of vacuoles. What is the meaning of devoid of vacuoles? Animal cells do not have vacuoles. Okay, so because animal cells are not having vacuoles, or because animal cells are devoid of vacuoles, in the case of animal cells, nucleus is usually located in the center of the cell. Nucleus is usually located at the center of the cell in the case of animal cells. On the contrary, if you come to plant cells, generally I am talking about a mature plant cell. In a mature plant cell, most of the volume of the cell, or most of the space inside the cell, is occupied by the vacuole. Most of the space within the cell is occupied by what? Vacuole. So because, now let us take a cell and most of the volume of the cell is occupied by the vacuole, so nucleus is pushed towards the periphery. It's not in the center, it's pushed towards the periphery, especially in the case of mature cells. So vacuole occupies majority of the place in the cell. So in the case of plant cell, the position of the nucleus, is usually towards the periphery of the cell. It is towards the periphery of the cell. In the case of plant cells, it is usually towards where? Towards the periphery of the cell. Okay, so this is based on where exactly the nucleus is located in the cell, whether it is located in the center or whether it is located in the periphery. In the case of animal cells, it is in the center generally. If you see some animal cells, especially in epithelial tissues, basal nucleus is also present. Nucleus is going to be present at the base of the cell. Okay, so those are not very common, but generally it is in the center, and in the case of plant cells, it is usually towards the periphery. Okay. Now let us look at the structure of this nucleus. Okay, so far we have been discussing about its location, position, etc. Now we will be focusing on the bigger picture, which is the structure of the nucleus, which is very, very important. Structure of the nucleus. Now, as we have already discussed, nucleus is basically a double membrane cell organelle. Nucleus is basically what? It is a double membrane cell organelle. Nucleus is also called as the brain of the cell because it is going to control most of the functions of the cell. So it is a double membrane cell organelle. So as soon as I say it is a double membrane cell organelle, this nucleus has two major components in it, or two major parts in it. The first one is the nuclear envelope. And the second one is the matrix, the fluid matrix that is present within the nucleus, and this fluid matrix which is present within the nucleus is called as nucleoplasm. Okay, so within this nucleoplasm, inside this nucleoplasm, if you see, you are going to have a spherical structure which we are going to discuss in detail, which is called as the nucleolus. Along with this, within this nucleoplasm, you also have the genetic material present in the form of chromatin. Okay, so basically if you look at this structure, it's a double membrane cell organelle. We'll see the structure using with the help of a diagram. It is a double membrane cell organelle. It's going to have a nuclear envelope. Okay, along with the nuclear envelope, you have a fluid matrix which is called as nucleoplasm. Within this nucleoplasm, you have a structure which is called as nucleolus and genetic material in the form of chromatin. Now let us look at the structure of this nucleus. Okay, so nucleus is more or less spherical in shape. So if you are looking at the structure of the nucleus, as we have discussed, nucleus is basically a double membrane bound cell organelle. Okay, so very, very important cell organelle. So it's basically a double membrane bound cell organelle. So I should be representing two membranes now. So what I am drawing is the nuclear envelope. Okay, I am actually drawing the nuclear envelope. Nice. So now what I've actually drawn here, I'm trying to draw the structure of the nucleus. Now this part is the nuclear envelope. It is having two membranes. Now this is the outer membrane. Okay, this is the what membrane? This is the outer membrane of the nucleus. What is this? Outer membrane of the nucleus. And this portion, this part, it is called as the inner membrane of the nucleus. Okay, be it any double membrane cell organelle, be it mitochondria, be it chloroplast, be it nucleus. This is common. Okay, the common thing is you have an outer membrane and you have an inner membrane. Now there is space. You can see the space between the outer membrane and the inner membrane. You can see the space between the outer membrane and the inner membrane. This space that is present between the outer membrane and the inner membrane, this space is called as the perinuclear space. What is the space called as? This space is called as the perinuclear space. Peri means around. What is the meaning of the word peri? Wherever we find peri coming, it means it is talking something about around. Now when you study about centrioles, there also a word comes which is called as the pericentriolar, means around the centrioles. Centrosomes, when we study, there a word comes which is called pericentriolar. Pericentriolar is around the centriole. Perinuclear, around the nucleus. Perimitochondrial, periplastidial. Okay, the spaces are named like that. So perinuclear space. So space that is present in between these two membranes, that is the outer membrane and the inner membrane, this space is called as the perinuclear space. Now, this perinuclear space is usually 10 to 50 nanometers. The size of the space is how much? 10 to 50 nanometers. Now, NCERT writes a wonderful statement here. NCERT states, this perinuclear space is actually forming the boundary of this nucleus. Okay, so this nucleus is separated from the cytoplasm. Now, this is the space that is present within the nucleus. This is the cytoplasm, the surrounding portion is cytoplasm. So, this nuclear envelope, this actually what? This actually together, this is together forming the nuclear envelope. This is together forming the nuclear envelope. Now, this nuclear envelope and especially the perinuclear space of this nuclear envelope is going to separate the contents of the nucleus from the contents of the cytoplasm. So, this perinuclear space is acting as a boundary separating the contents of the nucleus from the contents of the surrounding cytoplasm. So, I am making a statement here. Perinuclear space separates. What is perinuclear space doing? Perinuclear space separates the contents of the what? Contents of the nucleus. It is separating what? Contents of the nucleus from the cytoplasmic contents. Okay? It is going to separate the contents of the nucleus from the cytoplasmic contents. Now, this is the first thing. Second thing is inner to the inner membrane. Now, this is the inner membrane. Inner to the inner membrane, here you have a small network of protein which is going to form, this is called as the nuclear lamina. This is called as what? Nuclear lamina. It is present inner to the inner membrane. Now, what is the function of this nuclear lamina? The function of this nuclear lamina is to provide support, mechanical support to the nucleus. Now, you can see and very well appreciate that this nuclear envelope is not continuous. This nuclear envelope is having some spaces in between them. It is interrupted. Nuclear envelope is interrupted at certain regions. So those regions where the nuclear envelope is interrupted, okay, those regions where the nuclear envelope is interrupted, these regions where the nuclear envelope is interrupted, these regions are called as the nuclear pores. What are these regions called as? Nuclear pores. So nuclear pores are those regions where the nuclear membrane or the nuclear envelope is interrupted. So those regions are called as what pores? Nuclear pores. Now how is the nuclear pore formed? You can see NCERT also writes nuclear pore is formed by the fusion of these two membranes. Now see, this is one outer membrane and this is one inner membrane. These two membranes are parallel. NCERT writes outer membrane and inner membrane are parallel and they fuse at a point, and the point where they are fusing, they fuse at a point which is resulting in the formation of what? Nuclear pore. So nuclear pore is formed by, or it is a small gap, it is a small space that is formed in the regions where the nuclear envelope is fusing. Okay, so here nuclear envelope is fusing, you can see, here nuclear envelope is fusing, you can see. So this is going to become what pore now? This is going to become a nuclear pore. So what is the function of this nuclear pore? Very important. Function of this nuclear pore is, this nuclear pore, I am writing the function of the nuclear pore here. The function of this nuclear pore is, this nuclear pore, or through this nuclear pores, material exchange. What material exchange? Movement of RNA, movement of protein, movement of RNA, movement of protein into the nucleus or from the nucleus into the cytoplasm. Into the nucleus or from the nucleus into the cytoplasm. So instead of telling these two, I can use the word bidirectional. Bidirectional, when I am telling, I mean that RNA and proteins can move out of the nucleus and they can move inside the nucleus as well. Okay, so now why does RNA that is synthesized in the nucleus go outside? For protein synthesis. And some proteins that are needed, for example, DNA replication has to happen. So for DNA replication that has to happen within the nucleus, we need an enzyme called as DNA polymerase. And you know enzymes are proteins except for ribozymes. So enzymes are proteins. So if DNA polymerase is synthesized in the cytoplasm, that cytoplasmic protein has to enter inside the nucleus for the synthesis of, or for the replication of the DNA. So bidirectional movement happens. So who is facilitating the bidirectional movement? Nuclear pores are facilitating the bidirectional movement of RNA as well as proteins. Very, very important statement in the NCERT. This is the thing. Next, let us move on. This is about the nuclear envelope. So what are the points that we have to remember about the nuclear envelope? First thing is nucleus as a cell organelle is double membrane. It has got two membranes. First one is the outer membrane, second one is the inner membrane. Together they are forming the nuclear envelope. Okay, for 12th standard level, together they are forming the nuclear envelope. If you go to higher classes, you will have other components also being added to the nuclear envelope. But for now, nuclear envelope, outer membrane and inner membrane is present in the nuclear envelope, point number 1. In between this outer membrane and inner membrane, you have a space which is called as the perinuclear space, whose size is around 10 to 50 nanometers. Third thing is, the nuclear envelope is interrupted at certain places, and those places where nuclear envelope is interrupted, those are called as what pores? Nuclear pores. Now, through the nuclear pores, what is going to happen? Exchange of material, like for example, RNA and protein is going to happen from the nucleus to outside and from the cytoplasm to the nucleus. Is it clear till here? I hope it is clear till here. Now let us move to inside the nucleus. Okay. So when you are getting inside the nucleus, the space or this matrix that is present inside the nuclear envelope, this is called as nucleoplasm. What is it called as? Nucleoplasm, or it is also called as karyolymph. I am erasing the board so that I can make some space to make some sentences here. to write some sentences here. So, nucleoplasm. But a nucleoplasm is also called as karyolymph. It is also called as karyolymph, or it is also called as nuclear sap. It is called as karyolymph, or it is also called as what? It is also called as what? Nuclear sap. Now within this nuclear sap, what exactly is this nuclear sap, or what is present in this nuclear sap? Pay attention everybody. This nucleoplasm is a transparent semi-fluid, and it is a colloidal, it is a transparent semi-fluid colloidal substance. It is a transparent semi-fluid colloidal substance that is present within the nuclear envelope. So this transparent colloidal semi-fluid substance, what is it actually made up of? But in this nucleoplasm, you are going to have, bit of chromatin is present, as I have already told you. Along with chromatin, you also have your nucleolus. Very fine, that also we have already discussed. Along with that, what is present? Along with that, you have a lot of nucleosides. What is the nucleoside? We have learnt it in biomolecules already. Along with this, nucleotides are present. Difference between nucleoside and nucleotide, we have already seen in biomolecules. Along with this, RNA is present. Of course, DNA is present, the genetic material. Along with this, you also have some ions. Which could act as cofactors. Ions are present. Along with that, you have enzymes that are involved in replication, like for example, DNA polymerase is present, RNA polymerase is present, which is involved in transcription. So, all these components are present within where? Nucleoplasm. All these components are present within where? Nucleoplasm. Okay. Now, let us start with the first one, that is the nucleolus. Okay. So, nucleolus is a small structure, a small spherical structure. I am just erasing it so that I can make some space for nucleolus. But because you have already seen the labeling, I assume that you have already learnt what is the outer membrane, inner membrane, and so this labeling I can erase it so that I can use this space for my nucleolus. Now this is nucleolus. So within the nucleus, you have a spherical structure. Within the nucleus, you have a spherical structure, and this spherical structure that is present within the nucleus is called as nucleolus. Now, pay attention. This nucleolus is a non-membrane bound structure. It does not have any membrane around it. So, because we are talking about non-membrane bound structures, let us see what are the non-membrane bound structures in the cell. First thing, nucleolus, non-membrane bound structure. Second thing, we have centrioles or centrosome, which is a non-membrane bound structure. Third one, ribosomes, which are non-membrane bound structures. Now, if the question is, what are the non-membrane bound structures, non-membrane bound cell organelle in common for both prokaryotes and eukaryotes, then the answer is going to be ribosome. Answer is going to be ribosome, because the centrosomes are present only in animal cells. Nucleolus is present in eukaryotic cells. So, if it is a common question for both prokaryotes and eukaryotes, then ribosomes. Now, if the question is something like this, the question says, what is the non-membrane bound cell organelle or non-membrane bound structure that is present exclusively in animal cells? Then the answer is going to be centrioles, okay, because they are non-membrane bound structures, okay. So, let us come here now. Nucleolus, it is a non-membrane bound structure. Nucleolus is what? It is a non-membrane bound structure, means there is no membrane around it. It is a non-membrane bound structure. Nucleolus is generally spherical in shape. Nucleolus is generally spherical in shape. Next, it is a non-membrane bound structure. Next one. Nucleolus, the contents of the nucleolus are in continuation with the contents of the nucleoplasm. The contents of the nucleolus are in continuation with the contents of the nucleus or nucleoplasm. Why? Reason is because, what do I mean when I am telling the contents of the nucleolus are in continuation with the nucleoplasm? It means there is no boundary around this nucleolus. So whatever contents of the nucleolus are there, they are actually within the nucleoplasm. So there is no clear demarcation between the this nucleolus and this nucleoplasm. So the contents of the nucleolus are in continuation with the contents of the nucleoplasm. That is the second point. The third point is, nucleolus is the site for, very important point, it is the site for ribosomal RNA synthesis. Nucleolus is a site for what synthesis? It is a site for ribosomal RNA synthesis. rRNA synthesis happens in the nucleolus. Okay. Now you know rRNA, ribosomal RNA is a part of the ribosome. Ribosomes are basically what? Ribosomes are basically ribonucleoproteins, correct? So ribosomal RNA synthesis is going to happen within the nucleolus. Okay. So because ribosomal synthesis is happening here, the cells which are actively involved in protein synthesis, if there is a cell and that cell is actively involved in protein synthesis, those cells which are actively involved in protein synthesis, if the cell is actively doing protein synthesis, it is doing a lot of protein synthesis, if the cell is actively involved in protein synthesis, I am writing protein as PTN. If the cell is actively involved in protein synthesis, in those cells which are actively involved in protein synthesis, two things. The number of nucleolus is more. Number of nucleolus is more. Second thing, they are large in size also. Large in size, and the number of nucleoli is more in the cells that are actively involved in what synthesis? Protein synthesis. Okay. Now, so this is about the nucleolus, which is also present in the nucleoplasm. Now we are going to look at something really important, which is really important is the chromatin. What we are going to look at is something which is really important, and this is called as chromatin. What is this called as? Chromatin. Now what is chromatin? What is chromatin? Now listen carefully. What is chromatin? Listen carefully. Pay attention, very careful. Students often get doubt what is the difference between chromatin, chromosome and all that. We will see. Not to worry. What we are going to look at is something called as the chromatin. Chromatin. Now what is chromatin? Chromatin is actually loosely, or it is an elaborate structure which is having DNA in it. Chromatin is an elaborate structure which is having what? DNA in it. Along with DNA, it is having some proteins which are called as histone proteins. Along with histone proteins, it is having one more kind of protein which is called as non-histone protein, along with RNA. So chromatin is a loosely arranged thread-like structure. If you just look at a ball of wool, when you look at a ball of wool, how is it arranged? It is just a loosely arranged structure. So that loosely arranged thread-like structure is called as chromatin. Now this chromatin, what is it having? This chromatin, now just let us take for example, this is your DNA. Let us take for example, this is your DNA. DNA is a double helical structure. This is your DNA. To this DNA, we have some proteins which are called as histone proteins, which we will be studying in 12th standard. we have some proteins which are called as histone proteins. So histone proteins are associated with this DNA, and when the chromatin, when the DNA has to be packed, when the DNA has to be condensed, okay, so along with these histone proteins you also have some other proteins associated with it, and these other proteins are called as non-histone proteins. These other proteins are called as what? These are called as non-histone proteins. And what are these called as? These are called as histone proteins. So DNA along with histone proteins, along with non-histone proteins, and RNA together you call it as what? Chromatin. Usually chromatin is a 10 nanometer fiber. 10 nanometer fiber. Diameter is usually going to be how many nanometers? 10 nanometers. Basically DNA is a 2 nanometer fiber. So when it is getting associated with histone proteins etc., it is a 10 nanometer fiber. Now chromatin will further condense. What is the meaning of further condensation? Just imagine thread, just a thread that is used in stitching. When you take out the thread, it is a very long thread, correct? It's a very long one. Now I'll just show you this example. Now you can see this one. This is a thread. Now it is not condensed. It is an open one. Now what I'll do is I'll start folding it. Now what I'm actually doing is I'm condensing it. You can see this thread or this wire that I'm having, I'm actually condensing this wire. So you can see I'm actually condensing it. Now you see when I've condensed it, its thickness has increased. It is easy to pack when I condense it, correct? So this structure is the chromatin structure. And the condensed form of the same chromatin using some proteins to pack it within the small nucleus. Second thing is, just not for packing, second thing is during cell division. This chromatin, if it is in the form of chromatin, separation of the chromosomes into the daughter cells is going to become difficult. But when you condense it, when you pack it into a tight, small, compact structure, separation of these chromosomes, separation of this DNA into the two daughter cells is going to become easy. So highest condensed form, you call it as the chromosome. And DNA along with histone, non-histone and RNA, you are going to call it as what? Chromatin. Clear? Till here? Okay. So this is about chromatin. We will discuss about chromatin in great detail just in a few minutes from now. Now one more important point I have to make here is the outer membrane of the nuclear envelope. The outer membrane of the nuclear envelope, this is the outer membrane of the nuclear envelope. The outer membrane of the nuclear envelope continues as endoplasmic reticulum. The outer membrane of the nuclear envelope continues as what? Endoplasmic reticulum. Okay. Now, this perinuclear space, this is your endoplasmic reticulum. Now, what is continuing as endoplasmic reticulum? What is continuing as endoplasmic reticulum? What is continuing as endoplasmic reticulum? Outer membrane. Outer membrane of the nucleus is continuing as what? Endoplasmic reticulum. Now, this perinuclear space is continuing as the luminal space. Perinuclear space is continuing as the luminal space of the endoplasmic reticulum. Outer membrane of the nucleus is continuing as what? Outer membrane of the nucleus is continuing as the outer membrane of the nucleus is continuing as what? It is continuing as the endoplasmic reticulum. The perinuclear space is continuing as what space? Luminal space. Now on this endoplasmic reticulum, ribosomes are present. On this endoplasmic reticulum, what are present? Ribosomes are present. So if ribosomes are present, tell me, is it going to become rough endoplasmic reticulum or smooth endoplasmic reticulum? Obviously rough endoplasmic reticulum, because it gives studded appearance, correct? Now this rough endoplasmic reticulum, I am telling ribosomes are present on this endoplasmic reticulum. Now what ribosomes are they? They are ATS ribosomes. What ribosomes are they? ATS ribosomes. Now, again an important question. Now all of us know ATS ribosome is going to have two subunits. The first one is your 60S subunit and the second one is your 40S subunit. What is actually bound to the endoplasmic reticulum? Larger subunit. The 60th subunit will be binding to the endoplasmic reticulum. To the 60th subunit, obviously smaller subunit will be coming and binding. So to the membrane, actually who's binding? 60th subunit is binding. Okay. So we will end it here today and we'll be continuing it in tomorrow's class. But before we end, let us see what all we have actually studied in today's class. So what did we study in today's class? We actually started with the double membrane cell organelle, which is called as the nucleus. Nucleus is also called as the brain of the cell. Now, why is nucleus called the brain of the cell? Because it controls many functions in the cell, because it is having the genetic material in it. It is having the genetic material of the organism in it. It actually controls most of the functions of the cell. Second thing, we have seen the nuclei based on the number. We have seen the nucleus based on the position. Central nucleus, animal cells. Peripheral nucleus, plant cells. We have seen it. Then we moved on to the structure of the nucleus. Structure of the nucleus basically has two things in it. First thing is, it is having the nuclear envelope. Second thing is the nucleoplasm. We have seen the structure of the nuclear envelope and we have seen nucleoplasm. Within the nucleoplasm, what is present? Within the nucleoplasm, we have the nucleolus and the chromatin. Along with that, other enzymes etc. are present. So this is the overview of the nucleus. So what are we left out within the nucleus? We have to see the structure of the chromosome, which is very, very important. Okay, 2021 there was a question on the structure of the chromosome in NEET. So we have to look at the structure of the chromosome and we will be continuing that in the next class. Thank you very much.